Welcome everybody to Fast Pitch 2023. This is part of the continuing AOBIO program, Angel, Organ, Life, and Bioscience. And we are so pleased that you're able to join us today as we hear 9 pitch teams present their concepts as part of this program. This is a program of the Oregon Entrepreneurs Network in partnership with the Oregon Bioscience Association, Oregon Bioscience Incubator, and OHSU. And now I'd like to have a word from each of the partners, starting with Cara Turano with OEN.
Hi, Jeff. Thanks, everybody. It's Cara Turano, and I'm the President and Executive Director at OEN. I know I tell people all the time it rhymes with Sarah. OEN is a nonprofit that supports entrepreneurial efforts around the state for the last 30 plus years. And we are really excited to be partnering with all of these folks to bring to you Angel Organ Bio. If you see a company that you are really interested in and have interest in being an investor, please go ahead and reach out to me. And we hope that you will be really supportive of these folks tonight and ask some good questions. Thanks so much.
Thank you, Cara. I'm a quick learner. And now we will go to Renee Miller at Oregon Bioscience Incubator.
Hey, everybody. I'm Renee. I'm the Associate Director at the OBI. We are a 501c3 nonprofit funded primarily through State of Oregon lottery dollars. And we not only provide private office and wet lab space in Portland, but we also help support entrepreneurs statewide with our educational programs, mentoring, and networking events. And we're proud to be partners and sponsors of AOBIO, and we're excited to hear from our 9 startups tonight.
Thank you very much. And now we shall go to Ann Carlson, who will speak on behalf of OHSU.
Thanks, Jeff. Hi, everyone. I'm Ann Carlson, the Associate Director for Technology Development and Licensing in the OHSU Technology Transfer Office. The OHSU Innovates is a collaborative network at OHSU whose goal is to help advance OHSU research, innovation, and entrepreneurship in order to benefit society. And we're enthusiastic about being a partner at this AOBIO event for the 2nd consecutive year. And we're extremely proud to see OHSU innovations making their way through the commercialization process and new startups being launched based on OHSU intellectual property. So it's an honor for all of us within the OHSU Innovates Network to work with the researchers and the entrepreneurs at OHSU and those in the broader entrepreneur community in Oregon and beyond. And we're proud partners of this event. So please reach out to us anytime if you're interested in learning more about the exciting technologies that are being developed at OHSU. Thanks.
Thank you very much, Ann. And now a pitch on behalf of the Oregon Bioscience Association. First, we'd like to acknowledge our platinum members for their generous support, including ABSCI, AbCam, Biogen, Biotronic, CAI, Genentech, Microsystems Engineering, Pharma, Skanska, Twist, Bioscience, and VWR. This year, the Oregon Bioscience Association will again host the Oregon Pavilion at the Bio International Convention, June 5th through 8th in Boston. Bio 2023 will attract over 14,000 biotech and pharma leaders for several days of intensive networking. The exhibit floor is 99% sold out. However, the Oregon Pavilion is 50% larger than 2022, so have your company join us in the Oregon Pavilion. You'll get branding, visibility, and access to Bio's one-on-one partnering system, where you can start now to invite and be invited to meet with biotech professionals at a reserved table during the convention. To learn more, please contact our own Julie Black. We'll put her email in the chat. So here we go. Fast Pitch 2023. We have 9 pitch teams in the order of Tonsil Tech, Vital Flow, Heliomics, Trace Biosciences, Stone Stable, Pros Biosystems, Promedics, Humero, and Ethichem Pharmaceuticals. The rules are fairly straightforward. Each pitch has up to 5 minutes. We will have a timer, and this timer is pinned here so we can see it. Once the timer starts, it will count down. And as the timer gets to 30 seconds, it will turn yellow. As it gets to 15 seconds, it will turn red. After each pitch, we will have a 5-minute Q&A session. Please post your questions for each team through using the Q&A tab. Please start posting those questions as soon as you think of them during the pitch. You need not wait till the end of the pitch. Put those questions in as soon as you think of them, and we will ask those questions during the Q&A section. So let's get started. Our very first pitch team is Tonsil Tech. Tonsil Tech, if you'd like to turn on your video. So you got your video on.
Perfect. Can you see me?
We can see you.
Very good. Okay, well, I will get started. I see the timers there. Set my own timer on here too. Awesome. Let's do this. Hello, I'm Sydney. I am the co-founder and CEO of Tonsil Tech, where we're developing a new category in oral care for tonsil stone removal. Our vision is to provide safe and effective tonsil stone solutions for at-home use and dental offices. We engineered our tools based on customer feedback and quickly iterated with designs in 3D printing. We started selling in July of 2021 on our website, tonsiltech.com, and have sold over 235K to date. In January of this year, we completed our transition to injection molding, which improves our margins and positions us for growth. Tonsil stones are the calcification of food, mucus, and bacteria that form in the pits of tonsils. Tonsil stones form on a recurring basis and have a pungent sulfur smell. They feel uncomfortable, and wow, are they hard to get out. If you don't have your tonsils and you've never heard of tonsil stones, that's really not a surprise, because in the late 1980s, doctors stopped performing routine tonsillectomies on children. So young people are growing up with this frustrating issue that isn't being addressed. Based on over 15 scientific studies of X-ray and CT scans from around the world, conservatively, about 10% of the global population gets tonsil stones. That's 800 million people. And the target market in the US is estimated to be over 500 million. Now, with no products designed specifically for tonsil stones, historically, people were forced to be creative and use things that they find under their bathroom sink, like bobby pins, water flossers, pimple poppers, and toothpicks, which are unsafe and frustratingly ineffective for this stinky job at hand. And that's why we founded Tonsil Tech, to address this growing problem. Our patent-pending TonsilFix tools were specifically designed for easy, safe, and fast tonsil stone removal. The 3 tips are the correct sizes for tonsil pit openings and common tonsil stone sizes. The TonsilFix tool is gently inserted into the tonsil pit and twisted. Then the stone is easily captured in that loop and fully removed from the mouth, which is a very important part of the consumer experience, because you don't want to accidentally swallow these things. Very gross. Now, our flagship product is the premium kit that we sell for $45, which includes 3 TonsilFix tools, a suction cut mirror light to help see the tonsils, safety wrist straps, and travel pouch. Now, our customers can painlessly remove tonsil stones in about 30 seconds instead of 30 minutes of frustrated throat jabbing in front of the bathroom mirror. So super happy about that. But most importantly, now our customers can finally get their confidence back by removing such a potent smell. Now, when it comes to competition, there are 2 types of solutions, proactive and reactive. Proactive, such as TheraBreath, adds costly mouthwashes and sprays to your already busy daily routine. Reactive options allow for immediate removal, which is where we're focusing. And our product is better because it's specifically designed for soft tonsil tissue, which means there are no sharp edges causing bleeding or small parts causing choking hazards. My co-founder, Jesse, and I learned we both have tonsil stones during our bioengineering senior class project at Oregon State University in 2017. After college, we both went down to the Bay Area to work at startups in contact lens manufacturing, pharma, and 3D printed medical devices. While in the Bay, we met Daniel Forbes, our co-founder of Tonsil Tech, and we founded in August of 2020. Daniel is an experienced digital marketing professional who also works at a Y Combinator startup as the first marketing hire and helped the company grow from 200K to 10 million in revenue in 4 years. Now, we primarily acquire customers through paid social and paid search, and we're expanding our organic presence and affiliate networks and email marketing strategy. As we look to expand, we launched on our website, tonsiltac.com, in July of 2021. And we also started selling on Amazon in February of 2023 this year after we completed our injection molding transition. So that was a big win. And we're working on launching to dental office pilots in Q4 of this year and launching into physical retail stores in Q3 of 2024 after sufficiently ramping up inventory volumes. Now we're working towards hitting 10 million in revenue within the next 5 years, and our goal is to reach profitability by 2025. You can see how the 4 different channel categories are added and then scale over time. Now, after winning the early stage competition at the Ben Venture Conference in 2021, we raised $185K from Portland Seed Fund, Oregon State University, and Onami. Now we're raising $1 million in 2023 for 18 months runway. For marketing, salaries, increased inventory, regulatory for the dental pathway, and our goal.
And I'm sorry, time is up. We can start the Q&A timer now. And please put your questions in the Q&A tab. And the first question is, and I will unmute you, is you can unmute yourself. We're going to give you a little more time, but the first question is, what was your sales growth 2022 over 2021?
So in 2021, we did 26,000, and then in 2022, we did about 180K, and we're looking to do about 400K this year.
Very good. Any more questions for the Q&A panel, please? Question here is, tell us more about the FDA Class 1 licensing needed for sales in dental offices. How long will that take? And what will the sales cycle look like?
Okay, so I'll take that one piece at a time. So we're currently selling as a general wellness category, which is why we started with direct to consumer. When we go into dental offices, we'll be a Class 1 registered dental hand tool. And so that process in total will probably take us about 6 months, potentially even less.
Okay. Do dentists currently recommend at-home removal, or do they prefer to do it in the office themselves? Do they seem willing to recommend this at home, or might there be hesitance on the part of dentists to encourage this at-home procedure?
Yes, dentists definitely want you to do this at home. This is a reoccurring issue that can happen weekly. Some people get it daily. Some people get it multiple times a month. So dentists do not want you walking in their office every week bugging them about this. ENTs don't want that either, and neither does your general practitioner. Because this is something that is so frequent, it really requires having an at-home solution and something that's more affordable than going to your dentist on a regular basis to deal with this. So they are fully supportive of an at-home method. And then for people who are having more challenges or people who have a really strong gag reflex, they can actually do different things in office to help suppress that. But it's really only for people who are having a really hard time dealing with it at home themselves.
Very good. What was the valuation cap on the 180,000K investment?
5 million.
Very good. We don't have any questions currently. We'll wait a couple moments to see if we get any more. Here we go. Will dentists resell the product?
Will you repeat that?
Will dentists resell the product?
Will they resell the product? I'm not entirely sure what they're asking by that, but if they're asking, will they sell it in office? That's one option, although dentists don't necessarily want to have to deal with the inventory pieces of having it on hand. Some do, some don't. It really depends. They're each an individual style of business and they have their own way of doing things. However, having an affiliate or promo code that their customers can then go use that gives them a kickback instead, a form of affiliate marketing is one way that we could do that. And dentists actually highly recommend that we work with dental hygienists in their office because they're the ones that are having those patient relationships. They see them every 6 months and they spend the most time with them.
Very good. Next question. What are your financing plans beyond this seed round?
Yes, so our goal is to get to the point of profitability, and then at that point, we'll do a priced round and we'll take a look at how much we'll raise at that point.
What will the $1 million seed round be used for, and what level of investment do you need after that to get to 10 million in 5 years?
Yes, so we're going to use that investment in order to spend that on marketing, getting into these different channels, being able to get into dental offices, making sure we have enough inventory for places like retail. There's a fair amount of money that we will need to have on hand in order to have that inventory piece there. And so after that piece, we're also going to work on salaries and go through that FDA process as well. One of the ways that we got to the million, we were originally looking at 800,000, but ever since we got into a retail program, which will be announced later this month, that's one of the reasons why we decided to bump it up to the million. We think we'll need that in order to move forward.
Very good. Any more questions? In that case, would like to ask Sydney, thank you very much for your presentation on behalf of Tonsil Tech and being part of the AOBIO 2023. Now, please turn your video off. I'd like to ask Luke Marshall of VitalFlow to turn your video on. And are you ready?
Yep.
Let's go then.
Alrighty. You don't need a cardiologist to check your blood pressure. In fact, you do that every time you go to the primary care doctor's office, and you can even do it at home. So why is spirometry, which is the true north metric for your lung function, stuck in the pulmonary lab with the pulmonologist or the respiratory therapy specialist? This situation is causing a bottleneck for the 40 million patients with asthma and COPD, say nothing of the 25 million that now have long-haul COVID. And the result is delayed treatment times with late wait lists up to 6 months, missed testing with only half of patients with these conditions ever having done spirometry, and reactive medicine. We have 3.2 million trips to the ER every year due to asthma and COPD alone. And between ER visits, hospitalizations, and hospital readmissions, that costs us almost $30 billion annually. So what to do about it is to simplify the process and put a pulmonologist in every primary care doctor's pocket. And that's what we do at VitalFlow. I'm Luke Marshall, founder and CEO of VitalFlow, and excited to dive in on what we're up to. How we do this is by making spirometry easy, flexible, and powerful. So we have a broad-based platform that starts by enabling spirometry through a mobile app. So this device coaches patients through and collects the data seamlessly and transfers that back to the clinician reporting dashboard with intuitive, interoperable data management to make this process easy for primary care as well as specialists. All of this is done with third-party, off-the-shelf, FDA-cleared devices that integrate into our system through a Bluetooth connection. It's important to make these easy and flexible because that helps promote regular testing and proactive monitoring. And we make it easy and flexible with real-time coaching, with automated feedback, as well as grading, so that the doctors know that the testing on the way in is good data. We have simplified the process, which actually saves about 75% of the normal clinical time for doing this procedure, making it more feasible to do in the primary care setting. And we use calibration-free devices, which saves at least 20 minutes of time on a daily basis just to have this enabled in the office. We also have a hybrid platform that enables care anywhere, both in office and at home with patients. We make it powerful through better interpretation and proactive care with a lot of data science tools that are financed through non-dilutive sources like the National Science Foundation. We've raised almost $2 million in grant funding to date to promote building the tools for patient risk forecasting, readmissions prediction, as well as disease phenotyping and other clinical decision support software. It works through a subscription model. We scale our subscriptions based on the number of devices deployed, whether in clinic or at home, at $500 per year per device. We also have a consumable component where we make about $500 per box of consumables, which represents about 100 tests in clinic. And this works for both us and the providers because the providers have about a 5x at least ROI per test that they do in the office using existing insurance reimbursement codes. We started in the market in the clinical research area, and we have over 1,000 patients enrolled in studies that we're being paid for. And we view this mostly as marketing that we get paid for, because as they start to publish these studies through peer review, we're going to use that in our other channels. In the beginning of 2022, we launched into the independent primary care market, and we saw a very rapid uptick in that market, growing 1.5x quarter over quarter throughout that year and into Q1 of this year as well. And through 2023, we've really been focused on growing our accounts, focusing on health systems. We closed our first health system account, Sova Health, in May of 2022. And actually, the representative there is our biggest fan. She'll get on the call with other prospective customers and help us sell them, including our newest customer, First Health, which we are launching 12 clinics for this week with our second health system customer. So after 1 year in market, we're now accelerating our growth, and that's really the focus of 2023. We're projecting to be at 1,000 subscriptions by the end of the year, representing more than $1 million in ARR by focusing on larger accounts, at-home monitoring, as well as getting a faster market through high trust certification. Got a great team, which I'll allude to in just a second. And we'll leave it there. Hello?
Hey, we're ready to go with the questions. We can start with the question timer. And the first question is, what's your IP and how do you protect it if the devices are off the shelf?
Great question. Our IP is all in the data science and the data management. We have a iPhone app that actually, it's iPhone and Android, all written in Flutter. That is a very specific workflow that helps do spirometry more quickly, more efficiently. There's a lot of automated coaching that is in that app that basically replaces a human that would normally be sitting next to the patient during this procedure, who is coaching them through how to do the procedure correctly. We've built that all into the app, and that is our IP. Through the data science piece, we have predictive models that can actually forecast next day symptoms. For asthma patients, we did that as a proof of concept study with UNC School of Medicine. And we have additional algorithms we're building under the hood through the NSF grant funding that we're taking through to market over time. Right now, everything that we elevate into the product is as clinical decision support software. Right now, we protect all this as a trade secret. Over time, we'll file these as patents as well.
Great. Next question. How do you keep the data transfer HIPAA, HIPAA compliant?
Yeah, we made everything HIPAA compliant from the ground up. We've done that through having a segregated model in our cloud. So there's actually no PII that is stored in the same location as PHI. So for the layman, that's identifying information is stored separately from health information, and they can basically be hashed back together through the interface. So we made sure to build that from the ground up so that we are straightforward. And that actually gives me a chance to talk about high trust certification, which is essentially SOC 2 type of third-party audit specific to healthcare. And one of the things that they will actually provide as a third-party audit is showing that we are HIPAA compliant rather than us just claiming it and demonstrating it through our security documentation. We'll have a third-party source, and that's why we're pursuing that this calendar year as well.
Great. Next question. What sort of profit margins are you expecting?
Great question. So for the first year when they've deployed a device, we have a SaaS margin that's a little lower than normal for SaaS because we have the bundled hardware component that we bring into that subscription. So year one margins are around 68% gross. Those move into typical SaaS of 80-85% margins by year two. For the consumable components, those usually run around 50-55% gross margins.
Next question, and apologies if I mispronounced it. There appear to be many spirometry devices on Amazon. What kind of development is required to integrate each device into the service?
Great question. Any device that you're going to find on Amazon is actually going to be a peak flow meter, maybe labeled slightly differently. That's a little bit of a nuance. The equivalent would be a peak flow meter is to spirometry what blood pressure is to an EKG. So there's a lot more data you can get off the spirometer. It's a prescription device. That's why we only sell to the providers. So if it's an over-the-counter device, it's non-competitive in terms of the data that's coming off of it already. There are other competitors in the market. The incumbents tend to be partners with us. As I said, we use off-the-shelf devices. So we have a couple different device partners that we integrate with. For other startups, there are companies like Alluna and NuvoCare that have more of a full stack approach where they have their own device and their own software. That has different challenges in terms of supply chain management and that kind of thing. We decided to get to market faster by using the commodity devices that are on the market and making them smarter through our software.
Okay, next question. If there's no calibration, how does a clinician know if the device is malfunctioning?
Great question. Basically, there is no malfunction in terms of the way that the data is represented. If there is a malfunction with these types of devices, they basically just don't connect. And so that's a very easy to identify issue. The reason why these are calibration free is because they use a different sensor technology. It's part of why we like them. They use an infrared sensor looking at a rotor. So everything is very straightforward, very easy to calculate. Whereas the traditional devices use a mnemonic sensor, meaning it's pressure-based. They're very finicky. They change with the weather, literally because they change with temperature and humidity. So they can be tricky to calibrate, and taking that off the plate of the primary care doc who's already overburdened in terms of time is a really important part of the solution.
Great. Thank you, Luke. Thank you for your pitch and for your answers. And if you could hide your video now, we'd like to bring on Frank with Keliomics. Frank, if you turn your video on and start your screen share.
Hi, everyone. Thanks for having us on. And are you able to see all this?
You're clear.
Okay, great. So I'm Frank Bao. I'm a surgeon scientist and associate professor, and also the CEO and co-founder of Keliomics. The technology underlying our platform came out of my lab. So the problem we're addressing with OncoScreen is that personalized medicine for most cancers remain moonshot. This means that treatment selection remains population-based, essentially making patients their own guinea pigs. And a great example is Taxol, one of the most commonly prescribed cancer drugs, only works well in about 23 to 56% of patients. Patients, on the other hand, suffer severe toxicities and side effects without knowing if the treatment will work. Healthcare payers spend millions per patient, also without knowing if the treatments will work. And typically, treatment failure is diagnosed only when the cancer returns. This is emotionally devastating, and recurrent cancers often develop resistance, meaning subsequent treatments become less likely to work. Our solution is OncoScreen. So in OncoScreen, the tumor sample, say in this example from a breast cancer, goes into our system. We treat dozens of treatments and combinations tested outside the body, so the patient suffers no side effects. And then based on the readouts, the patient can get the best treatment for her cancer. And the fantastic news I have to share is that OncoScreen is in clinic. Our first breast cancer patients have been enrolled in our pilot study at Legacy Park. What makes us different is that for over 40 years, multiple technologies have tried to solve this very same problem. None have made it to clinic. The reason is that they've only in the past looked at cancer cells, and this doesn't predict response to therapy. Our technology is the only one that works with tissues and not cells, and I'll explain this right now. So this is a breast tumor, that's the red lump in the middle. Prior technologies focus just on the cancer cells, so they would take a tumor or a piece of it and try to take the cancer cells out to study them or test them. But we know that tumors are comprised not just of cancer cells, but also stromal cells, immune cells, and all these other cell types that influence it. What we're able to do is keep the entire tumor alive. And not only that, but also what we call the tumor microenvironment, meaning the fat cells, the breast ducts, even the bacteria that might be around that tumor. And this means that our technology provides the maximum accuracy in terms of predicting response to therapy. And the novelty of our approach is confirmed. We have multiple patents in the United States and Germany that were granted, and additional ones which are under review right now. The market opportunity for OncoScreen is large. The primary customer will be insurance companies because they're the ones who pay for these types of treatments. We've spoken with them and validated our pricing, and we know that we can comfortably set our price at $20,000 per test. So in the 4 types of cancer that we're addressing first, breast cancer, prostate cancer, colorectal cancer, as well as what are called peritoneal metastases from things like ovarian cancer, we have total addressable markets of $15 billion a year. We know that all the stakeholders key to the adoption of OncoScreen are aligned. Doctors and patients want better cancer outcomes with fewer side effects. And thus far, we've had 100% clinician buy-in. Payers want to spend less on expensive treatments that don't work, as well as on treatment failures, which means spending on palliative care. And pharmaceutical companies are able to report higher efficacy because OncoScreen can effectively convert all treatments into targeted therapies. From a regulatory perspective, FDA approval is not required. Instead, we are regulated as a lab-developed test. And so we function under GCLP, CLIA certification, and essentially we are regulated or overseen by Centers for Medicare and Medicaid. Our team has, collectively, we have more than 30 years of experience with entrepreneurship, business development, services development, clinical trials, payer reimbursement, marketing, and sales. So we have a full C-suite, and together we've had over 3 exits, completed more than 16 clinical studies, participated in 8 startups, and raised more than $25 million. We have a world-class advisory board, including Colonel George Peoples, who runs the largest breast cancer clinical trial organization in the country, Dr. Nathalie Johnson, who is this year's president of the American Society of Breast Surgeons, and one of our biggest champions in the legacy in the Portland area healthcare ecosystem, as well as others, and that I'll skip over in the interest of time. We've seen significant traction and awards, despite having only launched our service, only built out our labs last November. So our first patients have already been enrolled at Legacy Health, and the pilot study for this looks at triple-negative breast cancer, HER2 cancer.
Sorry, Frank, your time is up. I'm going to ask you to, are you back with us now?
Okay.
Okay, and we are going to go to the Q&A for this. I don't see any questions yet, but please submit your questions to the Q&A panel. Here we go. Do you plan to leave academia to focus full-time on this or find someone else to run the company?
Great question. So as of April 1st, so last week, I've scaled down to 70% in academia. And as our revenues and funding ramps up, my plan is to go to full-time with Chiliomics by end of year.
Next question. Could you clarify product pricing and who pays for it?
So as with most things in healthcare, it's the healthcare payers who pay for it. In terms of product pricing, the average new cancer therapy is $100,000 per year. What we're seeing time after time is that in phase 4, so post FDA approval monitoring, many of these new medications are not as effective as they were kind of sold to be during the FDA approval process. So what we are able to do is help insurance companies prevent spending on drugs that don't actually work for any given patient.
Thank you. Next question. Please tell us your current funding status.
Yes, so far we have raised $1.9 million out of a $7 million seed round. This is convertible debt.
Next question. What is the design of your pilot trial?
So the pilot study is similar to other pilot studies that have attempted to do the same thing. So it's 10 patients with triple-negative breast cancer, 10 patients with HER2-positive breast cancer, and 10 patients with advanced ovarian cancer.
Next question. Can you describe how the platform identifies the targeted therapeutic for the given patient? In parentheses, sample.
Yeah, that's a fantastic question. So the readout we're using is the same as what is used clinically, right? So typically patients who are getting what's called neoadjuvant chemotherapy. Once they've completed that therapy and they've seen a response, they'll go and have their tumors removed. Those tumors are then sent to pathologists for interpretation, and they use a grading scheme called pathologic complete responsive therapy. They're basically determining, the pathologists are determining to what degree the tumor responded to therapy. Because our system is all in vitro, but we are able to keep the same tumors alive, we will send those tumors to the same pathologist for interpreting the primary human tumors, and we're doing a one-to-one correspondence or comparison with the treated in vivo tumors.
Next question. What strategic pharma have you spoken to, and are you working with any of them yet?
So our technology is really a platform, and there are numerous applications. In our excess bandwidth as we're ramping up OncoScreen, we are starting to work with drug developers, and so we are about to sign 2 academic labs who will be revenue generating customers to use our technology to determine if their medications work. And additionally, I'm on my way back from a drug developers conference in Texas, where we've gotten promising leads from 7 different oncology startups.
Next question. How is this screen informative in terms of potential side effects when it's only testing the tumor tissue? I thought side effects are negative effects of the drug on the healthy tissue.
Yes. So the side effects we're able to avoid are the ones produced by therapies that don't work, right? So the least desired scenario is for a patient with cancer to get a chemotherapy regimen. Turns out it doesn't work on the tumor at all, and they have to try another one. Sometimes that doesn't work, and then they're on to third-line therapy, and maybe that's the one that works, right? So what we're able to do is test all 3 therapies outside the body and say, okay, the first 2 actually were not at work. It's the third one that would work. And the patient can then skip that cycle of treatment and side effects and finding out if they work.
And next question, what's your exit strategy?
So we are looking for a Series A after a successful pilot study, because successful pilot study will be a major inflection point. After that, we'll scale nationally, and somewhere in there we had to do an IPO.
Great. Thank you so much, Frank, for telling us all about Heliomics. Please turn off your video and mute yourself. And now we'd like to bring on Connor with Trace Biosystems.
Okay, let me share my screen. All right, can everyone see that?
It's good to go, then let's start the timer.
All right. Hi, everyone. I'm Connor Barth, co-founder and CEO of Trace Biosciences. We're a biotech startup creating imaging agents for surgery that specifically highlight nerves. I'm a scientist by training, working in the field of medical imaging. You really see the benefit imaging has provided to diagnosis of disease, preoperative planning, but very little has been done to apply this powerful technology to treatment or surgery itself. So really our mission is to bridge that gap and use the power of imaging to eliminate surgical nerve damage by applying imaging to surgery itself. Surgical nerve damage is a remarkably common problem for surgery that ruins lives and causes lasting pain and loss of function. Imagine you're diagnosed with prostate cancer and are forced with the difficult decision to undergo a prostatectomy surgery as cancer treatment, where high-risk nerve damage can leave you with chronic incontinence impotence that cannot be repaired. This problem is faced by 25 million patients worldwide annually, and in total, 108 million patients are at risk for nerve damage following surgeries in all specialties. The root of this problem really lies in the inability to directly see nerves during surgery, where surgeons are forced to use landmarks to approximate nerve location, imparting really enormous stress on the surgical team and leaving patients with injuries that significantly degrade quality of life. Our solution, Nerve Trace, uses nerve-specific fluorescent contrast agents that allow surgeons to light up the nerves. Using this technology, surgeons can directly visualize nerves in real time with high contrast using clinically available imaging systems. As you can see in the video, our contrast agents utilize near-infrared fluorescence, giving our technology a major advantage in allowing the surgeons to clearly visualize nerves with high contrast and resolution that are buried beneath tissues or blood, even providing the ability to see the unseen and identify nerves that are being cut in an incision, as you can see here. This technology will really allow surgeons to save time to identify and spare nerves, improve outcomes by reducing nerve damage, and reduce costs associated with follow-on treatment and pain management. Our target market, prostatectomy, is a high volume procedure involving removal of the prostate for prostate cancer cure, where up to 60% of patients experience nerve damage, causing incontinence and impotence, and hospitals and clinicians are really hungry for a solution. Importantly, 80 to 90% of prostatectomies are completed using surgical robots with fluorescent imaging system compatibility with our technology. And this really enables that integration into the surgical workflow. Beyond our target market, NerveTrace could improve surgical outcomes in every surgical specialty. And I've listed a few high volume, high risk surgeries here. Importantly, we validated our technology works in the relative anatomy and surgical nerve models of these procedures. And with a 12 to 15,000 installed base of compatible imaging system in ORs right now, we estimate a total serviceable market of approximately $10 billion, where NerveTrace could become standard of care for surgery. This technology we build as part of the surgical procedure, where our main customers will be hospitals and payers. We'll target high volume surgery centers initially, where the technology can be easily integrated in those imaging systems that are already implemented. In following adoption by these high volume centers, we'll establish separate reimbursement approval and expand to smaller community-based centers. And all in all, this will provide customers value as patient care is improved overall through reduced surgical complications due to nerve damage, resulting in reduced OR time, reduced length of stay in the perioperative arena, and reduced costs of follow-on treatment and pain management, as well as just easier, more accessible precision surgery to improve the quality of care across experience levels and surgery centers. So far, we've raised $3.8 million, most of which is non-dilutive grant funding, to complete the CMC manufacturing, regulatory planning, and pharmacology and toxicology testing needed for IND approval. We anticipate starting phase 1 clinical trials by early 2025. And following IND approval for first in human clinical trials, we'll complete really an accelerated clinical trial plan for FDA approval as a new drug. And these plans are guided by communication we've had with the FDA, as well as our regulatory consultants and CROs. We anticipate a potential exit following phase 2 or phase 3 clinical trials, and are already in communication with several potential partners or acquirers who could be surgical device or imaging system manufacturers, contrast agent manufacturers, or mid-sized fluorescence-guided surgery companies. We're raising a seed round to complete phase 1 clinical trials and really build a strong foundation for rapid clinical translation. We're a team with a high level of expertise in this field and advisors that have helped us along the way, as well as our funding sources from both public and private sources. Thanks for your attention, and yeah, I'll take any questions.
Great, thank you. And we'll get the question timer started. And the first question is, how has your product development advanced in the past year?
Yeah, great question. So one of the things we're working on is integrating this and making sure that it works well with the clinical imaging systems. So what we've done in this preclinical phase is both done a lot of testing on imaging systems that are present. So for instance, one of the ones that's used most often for prostatectomy is the Da Vinci surgical robot, and they have a Firefly fluorescent imaging system. So we've collaborated with them in large animal studies, as well as in human nerve specimen staining, to show that it works in human nerve tissues. It has that cross-reactivity, as well as works with their imaging system and works in the workflows that are needed for prostatectomy. Additionally, we've had some significant communication with the FDA. We've had a pre-IND meeting with the FDA, as well as completed CMC manufacturing of our first agent, and are moving forward now with the IND enabling pharmacology and toxicology testing to get IND approval, and just putting together the whole IND package for first in human clinical trials.
Great, next question. How is the dye introduced to the surgical site, and how is it different than other neurovascular fluorescent dyes?
Yeah, that's a great question. So the way we're developing it is as an intravenous administration. And so this will work, is something that can be administered at the time of surgery, just before surgery, or during surgery to a main line. And then it distributes to the nerves over time. It's a small molecule fluorophore, so that's actually a main differentiating factor over other agents. And so that enables rapid pharmacokinetics to highlight the nerves and get contrast within about 30 minutes after administration. And that contrast remains out to about 4 to 8 hours after administration. So it works with most of those clinical or those surgical workflows that we've been seeing.
Great. At this time, we don't have any open questions. We'll wait a few seconds to see if any more questions come in. Okay, it looks like we don't have any more questions. So thank you for your presentation on Trace Biosciences, and please mute and turn off your video. Next up, we have Ken Stedman with Stone Stable.
Actually, Jeff, it's going to be Kabir from Stone Stable.
Thank you for the reminder, Kabir. You'll be Kabir Bhatia with Stone Stable.
Not a problem. Everyone see my screen? I mean,
You're in preview mode.
yeah, that's fine. I'm just
Okay.
trying to get everybody oriented here.
Okay.
Timer. Okay, I'm good to go.
Oh, great.
Thanks, Jeff. Great to meet you. Great to see everyone. And very excited to have this opportunity. We are Stone Stable, and we're really excited to present again from the last AOBIO. We made a lot of progress. We made a couple of pivots, so we're really excited to share this. We are currently focused on the cell and gene therapy space, which are a game changer from a medical innovation perspective. Just to talk a little bit and frame sort of where the cell and gene therapy space is currently, there are only about 24 FDA-approved cell and gene therapies out there in the marketplace, and the cost of those therapies are pretty large. So there's a slide here from Bluebird Bio's presentation where they justify the cost of their therapy at $2.5 billion based on the lifetime value for a patient who would require blood transfusion for the rest of his life. But to put that in reference, there are about 3,700 therapies that are under development, 1,500 of which are in preclinical, and 40% are cancer therapies. So this is a very nascent space with a lot of opportunity. I'm asked to go into presentation mode, so I'm going to go there, see if that works. Everyone see my screen?
Yes.
Okay, good. So one of the critical elements that's important for cell and gene therapies is they are currently serving orphan drugs at the moment, orphan diseases at the moment. And as they look to scale towards more mainstream diseases, that cost is going to have to come way down. Two components to that cost. One is the current gene transfer is relatively inefficient, and the viral vectors that are a basis for conducting that gene transfer can be really expensive. We believe that our silica coating process makes that gene transfer process 10 to 30 times more efficient, and silica is way more cost efficient than some of the viral vectors that are used. And so we will be one of the critical elements for scaling this technology. We have also created a platform technology, and we're very confident that our technology can work across a number of the different viral vectors that are used. Happily, about 50% of the gene and cell therapies that are being developed are using adenovirus, which we have a lot of experience with. We've spent the last year talking to a number of pharma partners, and we've gotten some really encouraging feedback around sort of the excitement around the technology and what they would like to see. And so we feel like we've got a blueprint on where we need to go first. We've got a few partners that we are targeting going forward with, and I'll come back to this in just a second. The competition for transduction enhancers actually validates the market. There are several out there already, but they are either really expensive, they don't work in vivo applications, or they only work for very specific viral vectors. Stone Stable works across all of those different elements. And one of the other interesting elements is Thermo Fisher is not on the slide, and we think that they would be very interested in a technology like ours, which is one of the reasons why we want to work with it. We're currently in the process of raising about $2 million in new capital, We'll use this for both in vivo validation, which some of our pharma partners have asked for, as well as securing some of the extending our IP that we have. We have currently 15 patents, but we would like to get more specific patents in the cell and gene therapy space, and then filling out our bench in terms of both technical team as well as a business development lead, so we can start to work with some of our pharma partners. Our path to revenue is a licensing model to an early industry research collaborations and then partnering with pharma. There are multiple elements that are going in our favor, which we're excited about, including the comfort that the FDA is getting with silica in clinical trials. We've got a really world-class core team. Ken's the founder of the company. He's a world-renowned virologist and extremophile. Ajay is has done a lot of pioneer work on using silica for coding viruses. And one of the critical things that we've done over the last year is assembled a world-class scientific advisory board. Thrilled to have Phil on, who's the co-founder of ABSci, John Beal, who's the CEO of Capsigen and developing gene therapies using AAVs, Mark Slivka and Derek Carter, who are both experts in their field. I'm going to stop there. Thank you.
Thank you. And we will now transition to our Q&A section. Please put your questions in the Q&A tab. First question, how much grant and investment have you raised to date?
Yeah, we've raised about $450,000 in cumulative dollars, and most of those dollars have actually gone towards securing our IP. We've gotten patents in about 150 countries. We've most recently raised about $125,000 in non-dilutive funding from a GAP fund and from Wanami.
Next question, what is your general timeline for approval?
Yeah, I'm guessing John is referring to FDA approval. And the way this works is for ex vivo therapies, the path to getting approval from the FDA is much shorter, because the cells are effectively, the gene transduction takes place outside of the human body. And as long as there's no viral vectors remaining, there are no issues with that. In vivo applications are in general further down the line. And what we're hoping to do is to get traction with some of the gene therapy companies and be part of their path to getting those the therapies FDA approved. So we don't expect that we will go through an FDA approval process in isolation. We expect that this will take place with a pharma partner with their drug.
Great. At this point, we don't have any questions in queue, so this is a great time. If you have a question regarding Stone Stable and what you've heard in this pitch, please put that question in the Q&A tab. We'll hang on for a few seconds and see if any more questions come down the pike. Next question. Have you partnered with any new research collaborators in the past year?
Yeah, it's a great question. We have. We're actually in the process of doing that right now. We've been partnering with Andre Lieber, who's a researcher up in University of Washington and the preeminent expert on adenovirus, and also one of the co-founders of Ensoma. And we're actually looking at we're testing their cell lines right now to see whether our technologies receptor mediated or not. We suspect that it is, or it isn't rather, that it isn't receptor dependent. And we're very interested in sort of where that research then goes from a commercialization standpoint, particularly with Ensoma.
Great. Next question. Will this be protectable internationally, especially against nation state actors?
Yeah, it's a great question. We have patents that are very broad in nature around the coding of viruses with silica, and we secured those pretty effectively with all of our broad claims in 15 different countries, including China and and India, where a lot of viruses get developed. And so we expect that will help us in the subsequent protections as well, but that's still to be tested.
Okay. What feedback have you received from VCs so far, and what do they want to see you do before they invest?
Yeah, it's a great question. So we've just started raising our $2 million fundraise once we had the data and we'd assembled a team. And so we're in the process of getting that feedback now. For a while, one of the things that we were trying to figure out was whether there was a market there. It was very encouraging to actually see that there's a fairly established viral enhancer market already. What's even more encouraging is that most of those enhancers only increase their gene transduction by 10 or 20%. So the fact that we're coming in with a 10 to 30 times improvement is going to be very reassuring. The other piece that VCs had looked for was whether we had traction with pharma companies. We have that as well. So we're going to test that out in the next couple of months.
All right. Next question, is your licensing agreement with PSU valid going forward?
Yeah, it's a great way to plug PSU. They've been a fantastic partner, and they have both continued to support us with the agreement that we have already, but also been very clear that technology that we develop within the halls of Stone Stable belong to us, and they have no claims to that technology.
Great. Thank you so much, Kabir, for representing Stone Stable tonight in your pitch. And please mute and hide your video. And next up will be Eugene with Pros Biosystems. Please turn on your video.
Okay, let me share. Okay, can people see my screen?
Loud and very clear. Looks like we're ready to go.
Okay. Good evening, everybody. My name is Eugene Tu. I'm the CEO and founder of Pros Biosystems. We're a biotech company that's creating a new protein sequencing technology for unlocking the secrets of the proteome. Our technology can run on any NGS or DNA sequencer. So why are proteins important? About 90% of FDA-approved drugs on the market today have a protein target. And while that seems pretty impressive, there's a huge untapped opportunity because only a small fraction of the proteins in the human proteome are actually well characterized and known. So most of the protein or proteome is in this dark phase or dark state. And that's because the technology today is geared towards detecting just what is known. So if you think of the human proteome as a dictionary, right now we're just dealing with the A's, and we don't know anything else about the rest of the alphabet. So we've developed a technology called PROS, which is protein sequencing by expansion, which takes a globular protein and basically just stretches it out so that we can run it on any NGS platform, NGS being next generation sequencing or DNA sequencing. What I've shown here is a nanopore as the preferred sequencer because we think that it's the most useful for pure discovery purposes because you don't have to label the protein. You can read it directly. It gives you results in real time. You don't have to know anything about the protein itself. Also, the nanopore is about $1,000, so you can get started with sequencing for really cheap. Other people have tried to do this and all have failed, mainly because it's very complex to sequence proteins because they have 20 different amino acids, which results in a lot of combinations and becomes computational, very challenging to decode what the sequence is. Also, trying to tame a sort of globular sticky structure and make it push through a 2 nanometer pore is something that's not designed for. This is, so figuring out how to make it work is the basis of some IP that we filed, and we have pretty broad coverage in that space, and we're continuing to mine it. Our first market is going to look at the proteomics core labs. So that's labs that do proteomics in terms of like old school Sanger sequencing or mass spec, which is the gold standard for protein analysis these days. If you assume, or if we assume 10% penetration, that's 1,600 labs, And we also assume that each lab has a pull-through of about 35,000 a year, which amounts to running 1 kit per week per lab. Then we come up with a gross revenue number of about 56 million in 2028. That's 2.5 to 3 years after our beta launch. That's since we're selling kits, essentially, we have margins in the 80–85% range. And this addresses a sort of a $40 billion total market in proteomics research. We have some competitors. All of them have some strengths and weaknesses. We're the only ones that actually meet the definition for de novo discovery technologies. Most importantly, we access an installed base of readers. So at the day of launch, we would capture 4,000 installed platforms that people can just buy a kit and run our assays. We don't have to use binders, so we don't have to know anything about the amino acids per se, and that also keeps the cost of the assays down. This is our team. We're all in the at OHSU. We're part of CEDAR and the CEDAR Accelerator with varying skills and experiences in entrepreneurship. I've run several startups with exits down in San Diego, so I'm also driving this one. What we're looking for is money for a 2-year runway. That amounts to about $6 million from our calculations. That's to hire more people, expand that team, hire some C-suite officers like a commercialization person, build out advisory, expand IP, and start to work on partnerships with reader makers. So since we are instrument agnostic, we've already talked to several DNA sequencer companies and already have elevated to the C-suite decision makers. So we hope to get some traction there. So I'll stop here.
Great, thank you. We can start the Q&A portion now and start the Q&A timer. And the first question is, do you have any external partners who have validated your technology?
No, not yet. We've only been working on this particular version for about 7 months.
All right, that's the total sum of questions at this point. Well, here's another one. How much funding have you received so far?
We have not received any external funding. We're funded by CEDAR, which is part of the Knight Cancer Institute, and a part of the Phil Knight Cancer Challenge money. And to date, that's amounted to about $450,000.
Next question. Are you looking for any NIH grants?
No.
Next question. Can you comment on throughput?
Throughput. So there's 2 definitions of throughput. One is how many samples you can run at a time. So the multiplexing capabilities of that. Since I'm have a DNA sequencing background, we can use a lot of the same sort of methods and techniques that people use in DNA sequencing now to multiplex or to do single molecule analysis. So that's one part of it. The throughput in terms of how many peptides we can sequence or proteins we can sequence, it is pretty scalable at this point, because if we need to run like a full human proteome or single cell proteome, we would just run the sequencers for as long as it takes to move all the sample through. And that could be, for a nanopore right now, that's about 1 to 2 days.
Great. Next question. Can you provide more details about, quote, de novo, unquote, protein sequencing? Can this system be used to, quote, discover, unquote, proteins from patient samples?
So the short answer is yes. What I mean by de novo is the proteins are heavily modified in all organisms. So there's a lot of post-translational modifications. And if you have to develop a antibody or some other affinity binder, to recognize what is a phosphoserine versus what is a tryptophan, then you sort of have to make all those antibodies. And if you look at the total number of PTMs that are possible in proteins, it's somewhere in the neighborhood of 300 to 400 different types. So that's a lot of antibodies that you have to cycle through. We don't do that because we are reading the amino acid directly. So whether it's a canonical amino acid or if it's a modified amino acid, we will see that signal in the nanopore or in one of the other sequencers. And then we just have to train the algorithm to recognize what that particular modification is. And we've already started doing our training sets. So we don't expect that to be a big bottleneck.
Okay, next question. Will there be any regulatory hurdles you expect, or is PROS considered strictly a lab process?
We're just a research tool right now, so no FDA is involved.
Okay, we have no pending questions. We'll hang on a few moments. Oh, here we go. Next question. How do you build the training sets and over what time?
We build the training sets by making contrived peptides and just sequencing those. So we know what the original sequence is, and then we train to those signals. So right now we train maybe 1 to 2 million reads per amino acid, and it takes about maybe a week to go through a half a dozen different training sets.
Okay, we've got about 36 seconds left. Just seeing if anyone is finishing typing up a question. Looks like that's it. Thank you, Eugene, for your presentation. Please turn off your video and mute. And next up is Scott with ProMedics.
Oh, looks like we left it on. That's good. Great. Good afternoon, everyone. I'm Scott Filer, CEO of ProMedics. I'm going to ask you to do something a little unusual, but first I got to get the slides to change. Hang on. Something's gone terribly wrong. Not sure why that's not working. Stand by. Let's try this again. Here we go. Hopefully this works. Good afternoon. I'm Scott Filer, CEO of ProMedics. I'm going to ask you to do something a little unusual this evening, and that's the panelists and everybody participating. I would like each and every one of you to pinch your finger. More specifically, I would like you to pinch your nail bed with your thumb. Now, what I want you to observe is when you press down, when you compress your nail bed, you'll notice that when you release, it goes from white back to pink. It blanches and then it recovers. Now, physicians have been doing this since the time of Hippocrates, and physicians do it today, every day, thousands of them, in order to assess patient peripheral blood flow. And impaired peripheral blood flow actually is a sign of a number of fairly significant pathologies. But what I want you to do is pinch it again, and I want you to time it this time. Was it 1 second? Was it 2 seconds for it to turn pink again? Physicians are making that guess every single day. And while they're trying to assess for that peripheral blood flow and the degradation therein, one of the most important related to that is sepsis. Sepsis kills more Americans than breast cancer, lung cancer, and prostate cancer combined. And it's also extremely expensive. It cost US healthcare nearly $27 billion with a B last year alone. A study published in Frontiers of Medicine by our friends at OHSU found back in 2020 that after 30 minutes in the emergency room, every 10-minute increment beyond that 30 minutes where a patient waits for blood lactate levels to come back from the lab is a corresponding $1,000 loss in operating margin. And that makes sense, given that the longer it takes to identify a patient with sepsis, the sicker they become. And the sicker they are, the more expensive it is to treat. Indeed, speed is everything. According to the American Journal of the American Medical Association, they found that for every hour that a patient is unidentified sepsis, there's a 12% greater likelihood that they're going to die. Now, one of the earliest physiological signs of sepsis is reduced peripheral blood flow. We interviewed nearly 200 ICU and emergency room physicians, and they all universally said that pinching a finger was one of their top 2 go-tos. But interestingly, nearly 95% of them said if there was a way to take it from a subjective observation and turn it into a dedicated digital measure that was objective, they would use it as a consistent patient vital sign. We were listening. Introducing FlowSense, a wearable, non-invasive, digital peripheral blood flow monitor for the bedside. It's already been used in 2,000 patient encounters under IRB at OHSU. And we just completed a 600 patient observational study wherein we were able to identify a sensitivity of 91% and a specificity of 82%. That puts it right in line with blood lactate. Only blood lactate takes on average 85 minutes, and ours can be done within 15. Also, reflective light, which is the basis of our technology, means that FlowSense is skin tone agnostic. FlowSense fits in the clinical workflow because it was designed by physicians in clinical workflow. It can be used to order tests, it can be used during patient triage, and for a first time in industry, it can be used to get a baseline measure and then trend to determine whether a patient's getting better or decompensating. We've raised nearly 1.3 million over the last 5 years in non-dilutive grants, in convertible notes, and in safes. And we were just awarded a $255,000 NSF STTR. We have 3 patents, and we finished our Q-sub and are on track for completing our 510K this year. We have a great deal of strategic partnerships as well as funders. And we're currently working on new prototypes with our friends at University of Portland. We have a number of market options. Most important, we can sell directly to hospitals. And we have already a sales funnel of physicians that are interested because they are already investigators using our device under IRB. And we've got great feedback. We've been working with a lot of MDMs lately, and we've gotten some good responses from them. Here's the team that'll get us there. I'm Scott Filer. I spent 10 years in bench-to-bedside innovation at Argonne National Lab. Steve, our Chief Technology Officer, was a former senior principal engineer with Welch Allen. David and Matthew, our co-founders, are actually academic physicians at OHSU. Bob Jenks was a former managing director for GE Equity, and Ravi is our image analyst who makes sure that our signal processing works. And I was going to have a great line about sepsis and partnering, but I think we'll stop here.
Great, thank you. Time is up, and we will transition to the Q&A portion. We do not currently have any questions in queue, so we will stand by a couple moments. First question, assuming your fundraising is successful, what's your time frame for going to market?
So actually, we are conducting a $300,000 bridge capitalization in order to complete our FDA clearance, which should be done by the end of the year. We expect to do a Series A immediately afterwards, in part to be able to manufacture our device and begin selling. We can begin selling within a year from now.
Next question. Does this have any additional applications outside sepsis?
What a fabulous question. Of course, peripheral blood flow and impaired peripheral blood flow are associated with a number of pathologies beyond sepsis. Whether that's hemodynamic issues, hypovolemia, or just general shock. There's already been 2 different types of utilization cases put in front of us. One is interventional radiology, and the other is in urology. So there are so many different cases. And in fact, we are at a high-level conversation with Loyola University Medical Center and EMS. They would like to have an IRB to conduct research to see if, one, they can earlier in triage identify sepsis patients while en route to the hospital rather than waiting to get there, and 2, whether they can monitor effectively peripheral blood flow while providing IVs for hypovolemic patients, so patients who have been shot.
Next question. Have you entered into any agreements with medical device manufacturers?
Wow. So yes, we have a non-disclosure agreement with one large medical device manufacturer, and the covenants of that agreement restrict our ability to mention who they are. And we are presently negotiating an NDA with Medtronic.
Okay, next question. Do you have data showing your device improves clinical outcome compared to standard of care?
Yes, I wish we could have David. He is in the attendees. Dr. Sheridan is actually here. He could tell you more specifically the case studies that justify stating that the answer is yes. Our latest 600 patient observation study did demonstrate conclusively that our test was as sensitive as blood lactate, except we were much faster, like I said, within 15 minutes, as opposed to the average of 85.
Okay, next question. If you get this 300,000 bridge round, is that a down round?
No, that is just funding for our current burn rate, and we would probably do it as convertible notes.
Next question. Tell us more about how your product will compete with Philips Caretaker.
Yeah, great question. So part of the reason we had a nice quote from our friend at Philips was that Caretaker actually is a pretty good complement with our device. We don't necessarily have to compete. If I was to show you our competition slide, it would demonstrate that actually we are the only peripheral blood flow monitor. The rest are all regular vital signs that are typically available within either remote patient monitoring or otherwise in hospital. Philips definitely has had conversations with us about what possible high-level soft integration might look like. So that's still a conversation underway. Because they don't actually have peripheral blood flow. They have everything but.
Okay, well, we currently have no open questions. We will just stand by a few moments and see if one pops up.
It's really awkward not having questions.
Well, actually, it looks like we might not have any more. So, Scott, thank you very much for your presentation on behalf of ProMedX. Please
Thank you.
mute and end your video. And we're now going to ask Alia with Humero, I'm hoping pronouncing that right, to share your presentation.
Yes, actually, that's perfect. The presentation mode. Wait, sorry, that's not right. How's that?
Perfect.
Okay, well, thank you. My name is Ailey Traer, physician scientist at OHSU, and I'm the CEO and founder of Humero. I'm happy to get a chance to talk to you about our product. So this is what we're trying to develop. We want to develop novel combination therapies to treat blood cancers. What we have is a scalable platform with proven clinical fidelity. And this is something we've been working very hard on to make sure that this translates to the clinic. And we also have traction. We've been working with a number of pharma companies. They're very interested in what we're offering, looking at both toxicity and efficacy of different drug combinations. And right now we're based in OHSU. We're in the accelerator program, the same one that Jean mentioned with the CEDAR program. And what we're looking to do is eventually spin out of this to the accelerator and create our new company. So acute myeloid leukemia is our beachhead. It's our initial focus. It's a disease that I know very well. I treat this in clinic, and it's a very difficult to treat disease. Patients often do well with initial therapy, but the relapse rate is extremely high, which means that there's a very poor prognosis because after a period of time, you tend to run out of therapeutic options. This leads to a 5-year survival less than 30%. Recently, there have been actually more new drugs, so we're actually getting a little better, I'm happy to say. But there's a lot of issues with toxicity. And as we try to put more of these drugs together, we're getting even more toxicity, which is creating a problem. So why is the bone marrow important? I think I think about the bone marrow all the time, but most people don't think about it at all. And this is just to show you an example of a patient that I treated with chemotherapy. The red line shows you, well, it's actually a targeted therapy, a pill. The red line shows you how quickly the leukemia cells went down in the blood. So actually, initially, after a week, I was thinking that things were going great. But a month later, when we got the bone marrow biopsy, you could still see that there were a significant number of leukemia cells still present in the bone marrow. And this is because of all the surrounding cells within the bone marrow. It impacts the response to therapy and is really critical for measuring response. And it also lasts longer. It takes about a month for us to get our traditional first response in the bone marrow. So this was the inspiration for looking at the organoid model. And so we really wanted to bring together the important components of the bone marrow so that we could understand how these leukemia cells were responding to therapy and not responding to therapy, and identify better ways to try to target them with combinations. We also wanted to build a normal model so we could understand toxicity, because toxicity is the other side of the coin in oncology. And if you don't have something if you have something that's too toxic, then it's not going to be effective for patients. So one of the reasons that many drugs fail in clinical models is because we've spent so much time on these failed trials with toxicity and not enough efficacy. And so for one drug going to being approved, it's estimated that it takes $1.5 billion, and about half of that is in failed trials. So there's missed opportunities. There's good drugs that are put in the wrong patients. There's also a lot of unnecessary toxicity that leads these drugs to failure. What we're doing is developing a platform to find the right drug combination for the right patients, saving time, money, and lives. And this is how we model toxicity. This is a normal bone marrow. So we can give a very toxic compound and it kills cells. That's easy. Anyone can figure that one out. But we can do it for a short period of time and then allow for recovery, which we can see about 2 weeks later. This is exactly what I see in the clinic when I give these types of chemotherapy drugs to patients. And all the models out there right now for the bone marrow can't model this recovery. So this is very unique to our platform, and one thing that pharma really is interested in. In terms of an AML model, our competitors also come up short. And usually this is because it's too short of a time period to look at. They look at a 3-day assay. And these are 2 patients I put on clinical trial. You can see both of them in the black bar. You can see we predicted they were going to respond, but on the left, the patient only had about half of their leukemia cells go away. And the patient on the right had almost no reduction. When we do the Humero model, we put the right components together. We can see we have a much better prediction of how these patients respond. We're engineering a platform so that we can use automated liquid handlers so that we can basically increase our throughput and time period and run this automatically without as much effort. And what we're looking for right now is we're at OHSU, we've generated a lot of interest, but we want to leap and create our first company. And we're going to spin out the toxicity portion first. And then we want to expand from there once we start generating revenue and do a Series A. And add our disease platform later. We also have a plan to include a clinical trial in the Humero. And I think that my time is up.
Great, thank you. We'll go to the Q&A portion, although at this point, we have no open questions. So please enter your questions in the Q&A tab. First question, does your model involve the use of any software?
No, this is all cellular. So we use primary human cells and not cell lines and put them together in a 3D matrix to recreate the bone marrow. And then we use, we use a flow cytometry, which is something we use in the clinic as well to measure response and effective drugs on cells.
Great. Next question. What makes your culture technique different from a standard bone marrow culture?
So most cultures just simplify and only have leukemia cells, for instance. We take the time to put together the stromal cells, the surrounding supportive cells, which keeps the leukemia cells alive much longer. So traditional assays, if you just put leukemia cells in culture, it lasts about 3 days. In our model, we can keep things alive for a couple of weeks, which is closer to the amount of time you would see a response from patients.
Great. Next question. What will the management team be after the accelerator? And do you have a full team, both science and business?
Yeah, so right now we're mostly science, and I apologize. I got my timer was off a little bit from the other timer, so I was but I didn't get to that slide. But we're mostly the science side, and what we're looking to do is to spin out and get business advice as well. So that's something we're looking to add.
Okay, we have no open questions at this point. We'll stand by for a few seconds. Next question. Have you had any external validation at other research institutions?
We haven't sent our platform out to other institutions to use it. There's a number of people who are working on similar types of models. There's always variations, but we haven't yet had anyone do exactly what we do somewhere else.
Okay, next question. What are the logistics or timeline from sample acquisition to results?
Yeah, so that's a good question. One advantage of our platform is that we can actually use frozen viable cells from leukemia patients, which is actually, that means, and we have a large repository of those samples at OHSU. So we have access to a large number of samples we can run on this platform. Now, the time to run it and put it through the assay is a couple of weeks, 2 to 3 weeks, which is a long period of time. However, it does give you a more accurate representation of what happens in a patient over a period of a month, which is when we measure clinical response. So it does take longer, but we feel actually that's one of our key advantages.
Okay, currently no open questions. We'll stand by for a few moments. Okay, it does look like we don't have any more questions. So thank you very much for your presentation. Please mute and turn off your video. And we're down to our final pitch, which will be with Doug with Ethicon Pharmaceuticals.
Everyone see the slides okay?
Looking good.
Right. Ethicon reformulates approved drugs with safety problems and then uses rapid approval pathways to get them to market. So Sativex contains THC and CBD in a 50% ethanol spray that's used up to 10 times per day to treat multiple sclerosis spasticity. So that's uncontrolled hand movements and other shaking. It's approved in 29 countries, but the alcohol spray is difficult to use, as you can imagine, if your hand's shaking, trying to spray it on the inside of your cheeks, tastes bad, and causes oral adverse reactions. Slide's not advancing here. There we go. So what we do is we reformulate the same THC, CBD ingredients into a Zydis oral dissolving tablet technology or formulation. This technology is actually used in over 20 drugs approved in 60 countries. What this formulation allows is for rapid absorption into the bloodstream to the brain. There's no ethanol to induce oral adverse events. It's much easier for MS spasticity patients to use a tablet on the tongue than try to spray something inside the cheeks or their mouth. It tastes good, and from a business standpoint, a very rapid time to market, 3.5 to 5 years, as opposed to 7 to 15 years for your more typical drug development path. We expect our product to displace Sativex on the market because it's higher purity, has much more convenient room temperature storage. There's no alcohol, and it's easier to use. And again, rapid time to market. Our pipeline will address large market opportunities. So our initial MS spasticity is a $2 billion market. Our next indication will be adult focal epilepsy, which is over a $3 billion market. And then our third indication on the list now is chronic cancer pain, which is over a $6 billion market. Our road to the market, as you can see here, is we'll approach Canada first, where we expect to get approval by 2026. We will use that data and information with our filings in the UK and Germany, followed by the rest of Europe. So getting approval in those territories by 2028. We will then take that data plus do some dose ranging studies for FDA approval, which we expect to get in 2029. Our management team has over 100 years of pharma experience. Collectively, we've had 4 successful exits, raised over $200 million, done acquisitions in the range of about 1.5 billion, and have successfully filed 24 INDs and NDAs. We will be commercializing our products through pharma partners, and then Ethicon revenues will come from licensing fees, milestone payments, and royalties on net sales by our pharma partners. In previous rounds, we raised $780,000, and our current $2 million raise, we've already raised $1.6 million. We are now looking to top it off with the final $400,000. These funds will bridge us to our $12 to $20 million Series A round, as well as support our GMP manufacturing for our clinical samples to run our bioequivalence studies that are needed for the regulatory filings. Thank you for your time and interest. I think I should be way ahead of schedule here.
Indeed, you are way ahead of schedule, so we can now transition to the Q&A portion. And first question is, are there regulatory issues around investment into Ethicon with THC still being a Schedule I drug?
In actuality, since we're already working with regulatory approved drugs, these THC CBD combinations in other countries, we will be first going into Canada, where there's already approved this exact same product, but we're reformulating approved in Canada, UK, Germany, and the rest of Europe. In our discussions with FDA, we would just simply have to do a dose ranging study with the combined products, since these are purified CBD and THC, not botanical extracts, and we're not actually using marijuana or hemp product itself. And we expect those to be done in 505(b)(2) type of regulatory filings.
All right, next question. Is your drug reformulation process proprietary?
Yes, in fact, first off, we'll be going into a proprietary oral drug delivery system that's protected by a large portfolio of patents from Catalent. As I said, there are over 20 drugs already approved in over 60 countries that are using this technology. We also expect that we will have formulations that may be themselves patentable as we work out what the actual formulation has to be of both CBD and THC, and at least our lead compound. The other mixtures will be for other indications.
All right, next question. THC is lipophilic, and the current formulation solubilizes it in ethanol, but you have a sublingual product. How does it cross the membrane?
So we have actually already done a dog PK study on sublingual delivery, and shown that formulation achieves comparable blood levels to the ethanol spray. You actually can do some formulations with some oils and aqueous phase, and that formula does work out, and we're just working out the details on that. Obviously, I'm not a CMC expert, so I can't get into the full technical details, but I can just say that we've already done dog PK studies and shown that we can achieve equivalent levels in the blood.
Okay, we currently have no open questions. We'll just hold on a few moments to see if some more come in.
Everyone's ready for dinner.
Oh, here we go. Next question. Have you thought about selling this in pot shops?
We are going for regulatory approved products, so these will be reimbursable from a health insurance standpoint. So these will be prescribed drugs with all the typical FDA regulated drug product quality and consistency. So no, we will not be doing over-the-counter pot shop type stuff.
All right. Well, we currently have no open questions. Again, we'll hang a few moments, see if one pops up. That appears to be all the questions. Thank you, Doug, for presenting on behalf of Ethicon.
No, thank you.
And that completes our lineup of 9 AOBIO pitch teams who have presented tonight. Thank you very much to all the pitch teams who participated. And thank you also to all the partners, Oregon Entrepreneurs Network, Oregon Bioscience Association, OBI, Oregon Bioscience Incubator, and OHSU. And at this point, I'd like to bring on Kara, who will say a few words about the next big event in this series.
Thanks, Jeff, and nice job to everyone that presented. That was really intense and super fast, and everyone did a really good job. And thanks to everybody that is out there asking good questions. This is officially kicking off our due diligence process, as one of these companies will receive an investment at the event you see on your screen right now. So Wednesday, May 17th, 3 to 7 p.m. at Amatera Winery. You can find out more information at the website on that slide. Also, you can always email info@oen.org, and we're happy to help. But that'll be a great event where we will have four finalists. So 4 of these 9 companies will move on to our next round. They will have a chance to pitch live. There will be an innovation showcase. You will see last year's winner give an update live from stage, and then we will announce the AOBIO 2023 investment. So we hope to see you there. And again, if you saw things you're interested in tonight and you want to join us on our investment fund, you can reach out to me again at info@oen.org. So we hope to see everybody there. And thanks again for your time this evening.
Great, thank you, Kara. And just also like to say a thank you to Renee Miller at OBI for handling the timer, and my colleagues Lisa and Julie at Oregon Bio for also helping out on the back end. And that completes this program. Thank you for coming and joining us tonight.
Right. Thanks, everyone. Good night.
Thanks.
Thank you.